Okay, so before I begin, I would like to thank the organizer for this event, and I would like to thank you for attending this talk. So today I will present you a method that show you how to use genomic annotation to increase statistical power when fighting eGenes. So this is the outline of the talk. So there are three parts. First, I will explain what are considered to be eGenes, and then what do I mean by statistical power in the context of eGenes. And lastly, I will explain to you how we can use genomic annotation to increase statistical power. So here's the definition of eGene. So eGene is a gene that contains at least one eQTL. eQTL is a SNP that is associated with the expression of a gene. So given this definition, it's quite obvious that fighting eGene is sort of very closely related to fighting an eQTL. And you don't need to fight, in fact, you need to fight more than just one eQTL. So the next slide here, I will explain to you what is an eQTL. So eQTL is an expression quantitative trait loci. So here's an example. For examples, you are given this data. This is the gene expression of many individuals. So here you have many genes. This is the SNP data. So suppose you want to find out if the SNP in red is an eQTL with respect to the gene in blue. What you would do is you fit a very simple linear model where Y is the gene expression and X is the genotype of the SNP in red. What you would like to do is you would like to find this linear regression coefficient beta, which is the SNP effects and the p-values of beta. So if the p-value is very small, that means that beta is very high. So the slope here would be very steep, so that individuals who have the SNP C would have a higher expression than individuals who have the SNP G. So here's the definition of eQTL and eGenes again. So here's an example. So this slide is important. So for example, suppose you have an eGenes, and you can see that so suppose you are testing 10 SNPs. You are testing 10 SNP with respect to gene A. So you have each of these SNP, you would compute the p-values for the linear regression coefficients. And this gene is not an eGene because the minimum p-value here is 0.3. And you can see that the eGene threshold p-values is 10 to the negative 6, which is a lot smaller than 0.3. So I have not described to you what is this eGene threshold p-value yet. I will describe to you very shortly. So for now, just assume that you are given this number for granted. So gene A is not an eGene, again, because the minimum 0.03 is a lot higher than this threshold. So here's another example. Suppose you have gene B. So the minimum here is very small, 1.5 times 10 to the negative 10. So B is considered to be an eGene because it has an eQTL. So the reason is because this number is smaller than the eGene threshold p-values for gene B, which is 10 to the negative 8. So if you look at this, these two numbers are not the same because the eGene threshold p-values, they don't have to be the same for every gene. So what is this eGene threshold p-value? So I will explain to you now. So this number depends on three factors. Obviously, it's dependent on how many SNP you are testing. It's dependent on the minimum p-value of the SNP in the genes. It's also dependent on the LD of the SNPs. And this number is computed by using the permutation test. So what is this permutation test? Well, this is a very straightforward procedure. So what you would do is you swap the gene expression among the individuals in the samples. Then you redo the analysis so that you get a different minimum p-values. And you would repeat step 1 and 2 many times. By doing step 1 and 2 many times, what you do is that you create a null distribution of the observed minimum p-values. So for examples, you would create a null distribution of this minimum p-value here. And by knowing the null distribution, you can easily get the, say, the top 5% quantile, which would then becomes your eGene threshold p-values. Okay, so now I will switch gear and discuss statistical power in finding eGenes. So statistical power is just the probability that you can detect an eGenes if the gene exists, if the gene is actually an eGenes. So here's a very simple illustration of statistical power. So let's consider the simplest case where you have only one SNP. Recall you are fitting this linear model when you are trying to find the SNP effects, which is beta I. So when you fit this linear model, you get an estimate beta hat, which is normally distributed with some mean beta. And here I'm using the variance to be 1. This variance does not need to be 1, but here for simplicity, I'm just using this as 1. So when you are testing for statistical power, you assume that the true mean is not 0, and you also assume a significant p-value threshold. So here I'm assuming that the threshold is 5%, which means that this black line here is 1.96. So statistical power, so the blue curve is the distribution of beta hat when the true mean is 0, so the mean here is 0. And the red curve is the distribution of beta hat when the true mean is not 0. So statistical power is the area under this red curve, which is on the right side of t. I'm using the function p as statistical power. So this is the statistical power for one SNP, but in practice, we test m SNPs. So we have many SNPs being tested. So in this case, statistical power become the average, the uniform average at every single SNP. So here you have this power per SNP, and here you're just taking a uniform average over m SNP. So if you are interested in this definition, you can read more about this function here in these two papers. Okay. So this is the function again. This is a very important function, and I will now discuss what is this 1 over m and what is this t. So let's talk about 1 over m first. So 1 over m is the prior probability that the SNP I is associated with the expression. In practice, some SNP are more important because SNP can be very close to transcription start sites, or it locates in highly methylated regions or DNA sites. So you probably would not want to use a uniform prior probability. So here's an idea. You can replace 1 over m with Ci, where now Ci is a unique probability that the SNP is associated with the expression. So you would have Ci higher than Cj if SNP I is more important than SNP j. And you would like to have the sum of Ci equal to 1, because Ci is a probability, so they add up to 1. So this orange box here, this is what we consider to be genomic annotations. So in this talk, this is what I consider to be genomic annotation. So for example, nearness to transcription start sites or DNA sites. In practice, you can consider this to be anything you would like it to be. So we have just replaced 1 over m with Ci, but we still have this t to play around with. So t is the significant threshold per SNP. And for examples, traditionally, if you want to control for false positive rates, you would use t to be 5% divided by m, where m is the number of SNP. So I'm using 5% because it's simple, but you don't need to use 5%. But if you look at this, we can be a little bit more inventive than using Bonferroni correction. So, well, the next thing we can do is let's replace T with TI. So TI would now be the unique significant threshold per SNP, and you want the sum of TI to be 5% to control for the total false positive rates. Okay. So this is the slide that summarizes what we've done that is different from what was used previously. So previously you have statistical power, which is just a uniform average of the power per SNP. But by using genomic annotation, you now have these two new terms, which is a non-uniform prior per SNP and a unique significant threshold per SNP. Okay. So now we have this function, and we want to make sure that statistical power is high. Well, it's very obvious what we should do now. We should maximize this function. So here we maximize this function with respect to Ci and Ti, because these are these two unknown parameters that we have. So in practice, if you look at this function here, if you jointly optimize Ci and Ti, it's a very complicated function. It's not convex. So what we do is that we assume Ci is given to us. So in practice, there are heuristic procedures for finding Ci's. For example, you can assume that if the SNP is in the DNA's binding site, then it can be 10 times more likely to be associated to the gene expression. So once you assume what CIs are, then finding TI is pretty straightforward, because now this optimization is a convex problem with respect to T. And you have these constraints on T so that they sum up to be alpha, and they are between 0 and alpha. Alpha is the false positive rate. So for example, you can have alpha equal to be 5%. So P is, this function P we assume to be a normal CDF. So now you solve this optimization problem to get the TI. So there are many ways to solve this, but I will not go too much into the detail of this math. Okay, so once you solve for the TI, you can do hypothesis testing. So this is now very straightforward. So you go over each of the SNPs that you are testing with respect to the genes. And if any of the p-value of the SNP is smaller than TI, then you consider the gene to be an eGene. And this is all you need to do. And so here we have the results where we apply this methods to the gene expression data. So here we test this method on the GTEx data, the liver tissue data. In the GTEx data, we have 97 individuals and about 21,000 genes. Here we only care about cis SNPs, which are SNPs that is a 1 megabase pair from transcription start sites. So here's the results. So I'm showing you this function again because this is a very important function which the methods rely upon. So here we assume that CI is 100 if the SNP I is within 150 kilobase pair from transcription start site of the gene, and it's equal to 1 otherwise. So this CI is given to you. So now the only task is to find TI, and once you find TI, you can do a hypothesis testing to see if the gene is an eGene. So we test this, and we find that out of about 21,000 genes, we find that by using transcription start sites as the annotation, we find about 1,000 more potential eGenes. So here are these two comparisons here. We also test this using several other annotations. So here the first three are the histone marks that relates to activations of the genes. And this is the DNA's binding sites, and this tells this is the transcription start site, and this is not using any annotation at all. So this column is the prior ratio. This means that, for example, if you if you have a SNP that is in the DNA's binding site, then the SNP would have the CI equal to 100. Otherwise, the CI is equal to 1. So because CI is the prior probability that the SNP is belong to the may affect the gene expression. So as you can see that by using these annotation, we find more potential eGenes than not using any annotation at all. So transcription start site seems to be the most important annotation as shown here. So by looking at this, you may wonder that, well, transcription start site may also contain DNA site, so there could be, a lot of confounding problems, right? So this is one of the potential future projects that I am working on. And so we have the software available at these two websites here. So this is the version, my personal version. So I think this is better to use. And so I would like to thank my advisor, Elizar Eskin, and these are the co-author of the paper. So this is the end of the talk.